webinar this afternoon is aware, we will be recording this presentation and we will upload it to likely YouTube and our website following this so that anyone who missed the opportunity to view can watch afterwards. We'd just like to thank you all for joining us and for signing in this afternoon. We'll be giving an update on our 2022 Connected Waters eDNA project, which is a collaboration between the SLELO PRISM program as well as the Adirondack PRISM as well. Rob, I'll let you give the first introduction.
Super, thank you, Brittany. Good afternoon, everyone, and thank you again for joining us today. My name is Rob Williams. I'm Director of Invasive Species Programs for the St. Lawrence and Eastern Lake Ontario PRISM. And I'd like to begin by saying that, by convention, early detection of aquatic invasive species typically relies on visual observation of the species causing harm or the symptoms thereof. Unfortunately, by the time a species is visually observed, the species has likely populated, making management of the aquatic invasives difficult at best. Genuine early detection confirms the presence of a species before it has the opportunity to reach the exponential growth phase of the invasive's curve and cause irreparable harm to the ecosystem of concern. Environmental DNA allows us to look beneath the water and detect invasives at their onset. We began using eDNA in 2016, and over the years we've learned much, and we hope to learn more, much more as this innovative technology develops. We're grateful for the support we receive from the New York State Department of Environmental Conservation Invasive Species Coordination section out of the Albany office. An important theme that advises our efforts is connectivity. And this connectivity approach also helps us to maximize our conservation impact. Another important theme is collaboration, for without which our efforts would not be as strong as they are. Our team, Brittany Rogers, Gabriel Yurden, and Brian Green of the Adirondack PRISM have advanced this year's eDNA efforts by identifying waterways that connect the Great Lakes and St. Lawrence River with inland waters along the western Adirondacks. And by identifying connected waters, we can increase our chances of making an early detection prior to experiencing the impacts. This in turn allows us a strategic advantage to prepare a response to identified situations. So please welcome our Aquatic Restoration Resiliency Coordinator, Brittany Rogers, our eDNA project specialist, Gabriel Yurden, and Brian Green, Aquatic Program Coordinator for the Adirondack PRISM. It's all yours, Brittany.
Thank you so much, Rob, and thank you everyone for joining. So I'm actually going to first pass the baton to Brian Green, if you would like to unmute yourself and share for a moment.
Hello, everyone. I'm happy to be here today and excited to share with you this project that we've been working on with the SLELO PRISM. And this is part of the APIPP, the Adirondack Park Invasive Plant Program's aquatic monitoring. And so when we really look at it, we break this down into our aquatic problem into 3 areas: prevention, monitoring, and then managing invasive species. And, our goal is to work in partnership to minimize the impact of invasive species on the Adirondack region's communities, lands, and waters. And I think it's really important in this project because we're working across the PRISM boundaries. And really, we put all these artificial political boundaries of counties and states and prisms on our maps, but I really like to think that watersheds are really the ecological functioning unit. They're the neighborhoods. They're really what connect it. And so working in these 5 watersheds that drain from the Adirondacks into Lake Ontario and the St. Lawrence Seaway is a really great frame of reference, and looking at where these invasion fronts are will be really helpful for us. So we're hopeful that we can do more of these cross PRISM projects, and hopefully this will serve as a model. And it's been great to work with Rob and Brittany and Gabriel on this. So thanks so much for everybody and all the hard work you've put into it.
Thank you so much, Brian. And sorry, everybody, for the weird tech issues we seem to be having. So for those of you who are not aware or have not met me before, I'm Brittany Rogers. I am the Aquatic Restoration Resiliency Coordinator with SLELO PRISM, and I'm acting as one of the project leads with Brian. So if you have questions for myself or Brian, we can put our emails right into the chat box so you can have access to that. And please feel free to reach out and connect with us at any point. I just wanted to briefly talk about the PRISMs and especially SLELO PRISM in case you're not fully aware of the PRISM network. I'm not sure where everyone's from or how you heard of this webinar, but we're very glad to have you join anyway. So the SLELO PRISM is the area along the St. Lawrence Eastern Lake Ontario region, and we work across multiple different water bodies and lands that are, as you can see from our boundary to the east, is very connected to the Adirondack program, which is kind of where we connected and really came up with this project idea. So within SLELO PRISM and the Nature Conservancy in general, our mission is to protect our native biodiversity and freshwater resources through our collaborative approach to invasive species management. And we're so excited to have this partnership with the Adirondack program and to be able to continue expanding our understanding of invasive species in our region. In SLELO, like other PRISMs across the region, we have some core programming that we focus on, including prevention, early detection, rapid response, management, and ecological restoration, as well as an important education and outreach initiative. We have many special initiatives that we focus on each year of different projects that we put our attention towards. And here you can see some of the highlighted aquatic programs that we are working in. And so we are always looking for new projects and guidance for ways that we can connect and collaborate with our other programs, including this eDNA monitoring with the Adirondacks. Megan, at this time, if you want to unmute and talk a little bit about the Pledge to Protect program.
Thank you, Brittany. So eDNA is just one of the many initiatives that SLELO PRISM has to offer. We've launched an outreach initiative called the Pledge to Protect, which is intended to engage and inspire you to take actions that are simple, positive, and impactful to protect lands and waters from invasive species. Next slide. So invasive species prevention and management is a year-round effort, but there are actions that you can take to protect your favorite hiking trails, paddleways, forests, garden, and community from the impacts of invasive species now and for generations to come. So to take the pledge, you can visit ipledgetoprotect.org, or you can scan the QR code on this slide and fill out the form. There are 5 environmental areas you can take a pledge to protect, including lands, trails, forests, waters, gardens, and communities. Each pledge category includes a virtual toolbox that you can explore on our website, or you can download a PDF version. And each toolbox provides you with easy to follow, impactful actions that you can take to prevent the spread of invasive species, along with resources to help you manage invasives you may already have on your property or in your community. When you take the pledge, you become a protector. And for each pledge you take, you will receive monthly emails that share simple actions you can take to fulfill your pledge, along with resources to help you become more acquainted with invasive species that you may encounter or the specific pledge or pledges that you've taken. So if you are interested in taking the Pledge to Protect and are interested in more than one category, choose the I'll Protect Them All option on the pledge form. That way you'll receive a single email showcasing resources shared for each category, rather than multiple emails. And after taking the pledge, you get instant bragging rights, collectible virtual badges, access to social media toolbox, and chances to win prizes. So I encourage you to visit ipledgetoprotect.org today and become a protector. Next slide. And another great way for you to get involved is to volunteer with us. There are a variety of volunteer opportunities you can partake in, such as assisting invasive species removal efforts, aiding early detection, assisting eDNA sampling and restoration efforts, or helping our watercraft stewards raise awareness of aquatic invasive species. You can scan that QR code to volunteer or visit our website. Thank you, Brittany.
Thank you so much, Megan. So I just wanted to bring everybody together onto the same page with a little bit of background on eDNA projects and our work in the SLELO PRISM. eDNA is more of a newer tool in what most scientists would call new in the last couple of years. So it hasn't really been around for a very long time. And many programs are still kind of up and coming and learning about eDNA and how we can use it. So in 2016 was when we first initiated our eDNA in SLELO PRISM, where we were assessing the feasibility of being able to use this as a tool. We had really great results from that project, which led us to a 2017 pilot project where we were collecting eDNA samples as well as using video surveillance. In 2019, we participated in a Great Lakes port study alongside with Finger Lakes and Western New York PRISM partners in Lake Ontario, Lake Erie, and the St. Lawrence River. In 2020, we really started to expand our program, and we started looking at 6 of our major inland rivers and water bodies within the SLELO PRISM. And we continued to expand that even further in 2021, where we started looking at more tributaries to both the St. Lawrence and Eastern Lake Ontario areas. This year, we're very excited that we were able to expand further into the SLELO PRISM as well as into the Adirondacks, thanks to our wonderful partners across our boundary. And Brian really kind of hit the nail on the head mentioning the watershed. So we do draw these boundaries that we work in or we recreate in, but then when you really come down to it and you look at it, everything is very connected. And so that's why this project really kind of is very important to our collaboration and also our understanding of the connectedness of our region. So without further ado, I would like to introduce our 2022 eDNA project technician, Gabriel Yurden, and he is going to take the floor for the rest of this webinar. And if anyone is interested in learning more about the history or seeing any of our reports, please feel free to visit our SLELO PRISM website and go to the eDNA page as well. So on to you, Gabriel.
Hello, everyone. It's great to be here with you all today, and thank you for that introduction from the rest of our team. So I'm very excited to share all of our updates from this summer's 2022 Connected Waters eDNA project. So as Brittany was mentioning, eDNA is a relatively new conservation tool. And environmental DNA is DNA that accumulates in the environment after being expelled by organisms, be it through feces, urine, reproductive byproducts, sloughing skin cells, etc. And eDNA can be very useful as an early detection tool for cryptic species, low density populations, hard to reach populations, which is especially applicable in our situation, trying to early detect our invasive species. And there's also is an efficient and non-invasive method compared to traditional surveying. We can cover large geographic regions, such as this study, covering such a large swath of northern New York and the Adirondacks, has a low ecological impact and a low impact on target species. We're taking a water sample and never laying eyes on or interacting with our target species. So the objectives for our study this summer, we were testing for 10 target invasive species in 5 watersheds. We're assessing the spread of invasive species between SLELO, PRISM, and the Western Adirondacks, and also working to implement eDNA as a conservation tool and support future conservation work using eDNA since it is an up-and-coming conservation tool still. So after this, I'll show a couple of videos that outline our sampling procedure. But a quick overview, we take 1 liter of water from each sampling site. It's filtered, the eDNA is filtered out and preserved and delivered to the lab, and qPCR, quantitative polymerase chain reaction, is used to analyze the samples. So I'll go ahead to those videos and hopefully they play smoothly. And I'd also like to say thank you to Alyssa Trudell, one of our boat launch stewards, for helping with some of our sample collections.
All right, so this is our sample collection part of the video. We're gonna first rinse off any decontamination residue that might be on the bottle when we decontaminate it in the morning. We're gonna do this 3 times. We're also going to do the lid. And then we have to collect our sample from upstream. We're going to collect exactly 1 liter of water. There you go.
So the gist of that video is that all of our equipment is decontaminated with 20% bleach solution, but we rinse each container 3 times in the medium that we're collecting from, and then collect upstream just to ensure that there's no cross-contamination, especially between sampling sites. And the next video outlines our filtration process.
Hello, welcome to part 2 of our eDNA sampling protocol. This section of the video will be the filtration process. So here we have the water sample that you saw collected in the last part of the video, 1,000 milliliters in a sterile 1 liter container. We have 1,000 ml flask and a hand vacuum pump. So first I'm going to connect the hand vacuum pump flask. We have a small funnel and a plug. Attach those. These have all been decontaminated prior to use and will be re-decontaminated after this round of sampling. Here we have a sterile filter and a housing they received from the lab that processes our eDNA samples. This attaches on top of the flask also. And we're very careful not to introduce any contaminants during any part of this process. So I'm gonna go ahead and start the filtration. I'll slowly pour the water sample into the filter housing and draw it through the filter with the vacuum pump. Depending on the turbidity of the water, it can take anywhere from 2 minutes to 20 minutes to complete the filtration process. And I'll continue to filter this sample, and then we'll come back to you with the preservation process.
All right, and one last video that shows how we preserve the samples after filtering them.
All right, so we finished our filtration process. We drew all of the water out of from the 1,000 ml container into the flask down here, and now we're left with the filter and its housing. So we have sterile forceps and a labeled vial that has a site-specific label. In this case, we're on the Oswegatchie River. So we'll remove the housing, and then we're gonna fold the filter 2 to 3 times onto itself. And we left the filter just a little bit damp so that it is easier to fold. We have that folded. I'll go ahead and open the vial, grab the filter by its corner, put it into the ethanol, give it a couple shakes to make sure that ethanol reaches the entire sample, and now we are ready to head to the lab.
So apologies if the video quality wasn't very good there, but hopefully that helped you to visualize the process a little better. As you can see, it is pretty straightforward. Our main concern is just to make sure there's no cross-contamination between sampling sites. And I'll just give a little bit of an overview of how the analysis works in the lab. I won't get too deep into it, but there are 2 species-specific primer replicates, primers that replicate a small section of the target DNA. And there's a binding site for a probe nested within each amplicon. And the probe contains a fluorescent reporter dye on one end and a quencher molecule on the other end. And the probe is cleaved during extension, separating the quencher molecule from the fluorescent reporter dye. And thus the reflorescence is proportional to the amount of template created. And on the right, there's an example for bull trout, and the bull trout DNA is replicating and creating that curve, the implication curve. And our partner lab of Dr. Nicholas Sard at SUNY Oswego who is doing that analysis and the qPCR of all of our samples. And yeah, the entire process is completed there, and we deliver the preserved samples to them. So I'd like to thank him for that partnership also. So as another portion of the study, at a lot of our study sites, I ran an ROV, or remotely operated underwater vehicle, and that helped us to just get a snapshot of the underwater ecosystem at some of these sites. And on the bottom, you can see some aquatic vegetation, a largemouth bass on the right, and that's a picture of the ROV itself up at Tupper Lake. And it's pretty cool. It can go a couple 100 feet out into the water column, go down to deep water with headlights, and really allows us to see some very unique ecosystems. So our target species for this study were, I'll start with the Asian carp group, and that's the silver carp and bighead carp. And there's same genetic analysis, same primer probe pair for that group, followed by the northern snakehead, tube-nosed goby, tench, rusty crayfish, and round goby. And then we had 3 aquatic vegetation species, being hydrilla, Carolina fanwort, and Eurasian water milfoil. And up on the right, that's actually a rusty crayfish that I caught at one of our sampling sites. So it'll be interesting to see if we have a detection there. So I'll give a little bit of background on these species and some of the characteristics that make them of concern to us. So for the Asian carp group, they're filter feeders that feed on plankton. They can dominate native fisheries in both abundance and in biomass, and they can consume 5 to 20% of their body weight per day, making them especially impactful. And bighead carp could potentially outcompete native fish and eliminate the main source of food for larval fish and native planktivorous fish. Native fish considered most at risk include ciscos, bloaters, and yellow perch. The northern snakehead, the juvenile's prey includes small prey such as zooplankton, larvae, small fish, and crustaceans. And these populations may be threatened by feeding juveniles. Adults devour fish, crustaceans, small amphibians, reptiles, and some birds and mammals. And so they're of special concern to us. The tube-nosed goby, it does not eat invasive zebra and quagga mussels like the round goby, so there's no added benefit there. And they also eat bottom-dwelling fish and compete with them for food, and compete with other fish species that spawn in nearshore areas of lakes and rivers. The round goby can take over prime nearshore spawning sites and aggressively prevent use by native species. They're prolific breeders, spawning every 20 days during the spawning season, and their long-term impacts are expected to include declines in native species populations. Rusty crayfish, these are opportunistic feeders. They often displace native crayfish, and they can harm fish populations by reducing aquatic plant density, eating eggs, and reducing invertebrate density as well. The tench, they are in direct competition with native species for food, such as benthic invertebrates, snails, and fish eggs. They're generous predators, and their feeding behaviors increase water turbidity. And because of that, they can have negative effects on water quality for native fish. For our aquatic vegetation species, Eurasian water milfoil plant. The plants are rooted at the lake bottom and grow rapidly, creating dense beds and canopies. Stem densities can exceed 300 per meter squared in shallow water, and they're very difficult to control mechanically or chemically. And early dense growth shades and outcompetes native macrophytes, and that results in decreased macroinvertebrate, native fish, and native waterfowl populations. And also decreased swimming and boating opportunities can result, and decreases in home values, etc. Hydrilla. Hydrilla invades deep, dark waters where most native plants can't grow, and it can block sunlight and displace native plants below with its thick, dense surface mats. It can decrease or eliminate feeding areas for fish and waterfowl, obstruct boating, swimming, and fishing, and water stratification can occur, resulting in decreased oxygen and even fish kills. Carolina fanwort can form extremely dense stands, can clog drainage systems and waterways, interferes with recreational activities and causes economic damage from that. And it can also outcompete native aquatic vegetation also. So these are our sample locations. On the right, you can see a map with each of our sample locations highlighted with a point, and the watersheds are color-coded. So the 5 watersheds we were working in were the Black Watershed, Oswegatchie, Grass, Racket, and St. Regis. And we also had 3 points along Eastern Lake Ontario and 3 species-specific sites, which I'll talk a little bit more about later. And within each of these watersheds, we sampled a variety of tributaries and water bodies, some being frequently visited boat launches on lakes and some backwater streams. And we did 2 rounds of sampling, so you can see the number of sample locations on the right, and each of those numbers would be doubled for 2 rounds of sampling. And the map on the right, the color-coded map, starting up on the right in pink is the St. Regis, the Racket next in purple, and the light blue would be the Grass. Next is the Oswegatchie, and then lower in green is the Black River watershed. So we did a lot of legwork before going out in the field to try to maximize our sampling trips, because as you can see from the prior map, we covered a large geographic region, which resulted in a lot of driving, which is a lot of man hours, also potentially large carbon footprint with all the driving. So we had 10 sample days total per round, and 3 of those days I camped in varying locations to decrease the traveling time, and 2 days were minimal traveling along Eastern Lake Ontario and Watertown. So we only had two days of full out and back driving up towards the Adirondacks in northern New York. And these are some of the campsites that I was at. The St. Lawrence River was one. I was in Higley Flow State Park, and also Little Wolf Campground by Tupper Lake. And I think that definitely was fun for me, and also really helped us get a lot more done and have a further reaching study with the time we had this summer. So I'll talk a little bit about each watershed and some of the characteristics, and also I'll introduce some of the pollutants or factors that are affecting these water bodies, because I feel that is relevant, because invasive species are also another factor impacting the overall health of these watersheds. So to start with the Black Watershed, it's over 1,200 acres, and the eastern half of this watershed is covered by the Adirondack Park. Some pollutants affecting it are atmospheric deposition, invasive aquatic vegetation, as in the study, urban runoff, agricultural, and chemical spills. So urban areas make up 0.9% of this watershed, and they include Watertown, Carthage, and Lowville. And the source for a lot of this information is the USDA Natural Resources Conservation Service, and they do rapid watershed assessment profiles, which have a lot of useful information on these and many other watersheds, if anyone is interested. And also all of the pictures included in these slides are sampling sites that I visited. And here's a map of those impaired and impacted water bodies within the watershed. So next is the grass watershed. It's over 404,000 acres, of which 43% is in the Adirondack Park. It has relatively few impaired water bodies, but some pollutants are atmospheric deposition, agriculture, and contaminated sediment, and urban runoff. And urban areas make up less than 0.3% of land use there, those being Messina and Canton. And I was going to give a little introduction on some of the principal land uses within these watersheds, but they are relatively similar. The black watershed is about 60% forest, and the others are 70+% forest. And for the Oswegatchie watershed, it's over 671,000 acres, 44% is in the Adirondack Park. Some pollutants affecting its waterways are atmospheric deposition, chemical leaks, sewer overflow, agriculture, resource extraction, and that was zinc. And its urban areas are 0.3% of its land use, those being Governor and Ogdensburg. So the Racket watershed, it's over 805,000 acres, and 75% of its watershed is contained by the Adirondack Park. Some pollutants affecting our atmospheric deposition, agriculture, failing on-site systems, and construction. And its urban areas are Potsdam, Messina, and Tupper Lake at 0.6% of its land use. And I also think it's pretty interesting to look at some of the pictures on the side and just see the varying types of water bodies that are contained within each watershed, and even the rivers themselves, how they're changing with the geologic formations, etc. And finally, the St. Regis watershed. It covers over 555,000 acres, 60% of which is in the Adirondack Park. It also has relatively few impacted waterways, according to this study, but some pollutants affecting our atmospheric deposition, agriculture, and contaminated sediment. And also, according to this study, by far the largest impact pollutant-wise on these water bodies was atmospheric deposition. And there are no classified urban centers in the St. Regis watershed. And so our three sites along Eastern Lake Ontario were the Salmon River Estuary, South Sandy Creek on the left, and the Oswego River on the right. And the three species-specific sites that I mentioned previously, these sites had documented populations of target species, and 2 samples were taken in the Finger Lakes region. So we'd like to thank New York State DEC, Michael Robinson, and Finger Lakes PRISM Kate Monticelli for collecting those samples. And those species-specific sites were Carolina fanwort and hydrilla. And we also had a species-specific site at Stony Creek for Eurasian water milfoil. So we used field maps as a collection/navigation mechanism. We had a downloaded map, and I used a Garmin Glow and an iPad to provide a location to the iPad so I could ensure that I was in the right spot at each sample location. And the points that you see were all linked to survey 123. So once I could confirm location, there was a link that would bring me directly to our sample survey. And we used survey 123 to collect a lot of background information at each site. Some of the metrics that we collected included the water body type, turbidity, flow habitat, substrate, surrounding land use, weather conditions, depth of collection, sample collection method, and many more metrics. And survey 123 auto-populated a lot of the information you could see in the left picture. We had a site-specific code for each location that identified the watershed and sample number, and also specific coordinates for each collection site. And you can see a bunch of the already collected samples over on the right for several of the watersheds. So a final progress update. Our Connected Waterways project had 66 total samples plus 10 blanks times 2 rounds of sampling, which was a total of 152 eDNA samples collected. And laboratory analysis is currently in progress. So we're really looking forward to seeing the results from this study. And as I mentioned, some important sources where for some of this information, they're the USDA Natural Resources Conservation Service Rapid Watershed Assessment Profiles for a lot of that watershed information. And that is based in, was sourced in 2010. And for more information on these invasive species, you could go to Western New York PRISM Invasive Species Profiles and also New York Invasive Species Information Species Profiles. And also the SLELO website has a lot of relevant information on these invasive species. So with that, I would love to take any questions, comments, reflections, or anything else you'd like to say about this eDNA project. Thank you all.
If anybody has any questions they'd like to ask, you can feel free to send those in the chat, or if you're interested, you can unmute yourself and ask out loud. And we'll just give people just a minute or so to type in the chat box, but I just wanted to really emphasize again the importance of the collaboration between SLELO and APIPP, and also many of the partners who have assisted us, whether it's through the lab and student assistance there. We've had volunteers out helping us with sampling, State Parks, DEC, and many of the other programs that are working within our region. If anyone's interested in learning more about our exact sample sites that we went to in any of these watersheds, please feel free to reach out to myself. Unfortunately, today is Gabriel's last full day with us, so he won't necessarily be reachable through this project after today. But I will just kind of pause for a moment. We'll mute here, and if anyone wants to ask questions through chat or send them, via email or anything else like that, we're more than happy to receive those.
Gabriel, we do have a question in the chat box.
I did see at several of the sites closer to the St. Lawrence, but definitely not further up the watersheds. There were 3 sites, I believe, that I was able to positively identify them. So those in particular would be interesting to see if we have positive results, but I didn't actually see any of the species that we were testing for other than those. Terrestrial invasive species, yes, definitely. And we did use Seek and iNaturalist throughout this study. So any animals that I could identify, I entered on iNaturalist, invasive or otherwise, and a lot of vegetation, a lot of plant species also I would identify on there. So it would be interesting to see the native versus invasive species that are surrounding those sample sites on land also towards the end of the study.
I see a question in the chat from Zachary. It's what other eDNA projects in the PRISM, so in the Adirondacks there are a couple of different projects going on with some of our partners. So groups like ASRA, the Osceola River Association, U.S. Fish and Wildlife Service, the Nature Conservancy, and a couple projects where we're going and doing testing, eDNA testing for native fish species. So some of the trout and salmon species that are coming off from Lake Champlain. And then I believe there are a couple of research projects that are going on where people are using eDNA to try to test for some insects. So hopefully, like we talked about in this presentation, it is a new and upcoming monitoring technique. So a lot of us are working on ways to figure out how does it work, how effective is it, how cost effective is it, what's the detection rates, and not just for aquatic species, but also for some terrestrial species. So there'll be a lot of new and upcoming research. I don't think this is a monitoring technique that is going to go away.
I agree with that, Brian. And thank you for answering that question. So Colleen, to your question, typically we have our reports finalized by November or early December. And so we do have a little bit more funding for some more sampling to occur. And once we get the results for our current samples that we've already collected, when we get positive hits or amplifications for DNA, we'll be able to respond to those and collect another sample and hopefully be able to confirm with multiple amplifications that there's presence there. And Colleen, I would love to connect offline with you and learn more about the project that you're working on in Messina as well and compare our results. And Lola, to your question, I don't have offhand an exact number for our per cost sample, but if you'd like to reach out to me afterwards, I can calculate that and share that information with you. All right, so with questions kind of slowing a little bit, I just wanted to say thank you again to everybody who joined us this afternoon and who has shown an interest in this project. As I mentioned, this is Gabriel's last day, so I do want to give a special thank you to Gabriel, who has spent countless hours creating driving plans, deconning and using bleach on all of our supplies and making sure that everything was prepared for sampling, and also spending excessive amounts of time in the Adirondacks and getting to see some really beautiful sample sites that many of us don't always have the time to get out to and see. If anyone has questions or would like to connect, please feel free to reach out to myself or Brian, or if you're interested in the Pledge to Protect program, Megan, and also our program director, Rob, as well. And with that, I would like to say thank you all so much. I'm giving you all 15 minutes of your day back. And so I hope you get outside and enjoy some sunshine wherever you are for those 15 minutes. Thank you again and have a wonderful day.
Hey, Brittany.
Hey, Megan.